Bio, doing a biostat seminar. David's not here today, so I'm stepping in for him. But we are going to continue the series. We'll have a break the first Thursday of May for the biostat faculty meeting, but then the next 3 Thursdays in May seminars. We'll call it May-I. And we're going to focus those 3 talks on topics related to machine learning. So Juan Pablo will be talking, Chun will come back and do a part 2 for the talk that he did. And then somehow I'm down to do one of them, and I know very little about AI, so I'll stumble through, or I may try to find somebody to help me say something more intelligent. So there will be 3 sessions about AI. Not all of them guaranteed to be intelligent. I think that's about it. Anybody have any announcements on anything? Okay. Well, we are super thrilled today to welcome Dr. Joycelynne Palmer, who is currently at City of Hope and the director of the Biostatistics unit there, but is a former PhD student in our epidemiology program, dating back to the late 90s, I think, when she first came. Graduated in 2004. One of our superstar students, and has gone on to really make a nice career, I think, at City of Hope. So we're thrilled to have her back and really eager to hear what you're going to talk to us about. So welcome.
Thank you. Thank you, Dr. Gotterman. So I believe that you all can hear me. He told me I didn't need a mic.
And the acoustics in here are not great. So,
okay, maybe.
Well, just if you want to just make sure.
Okay, talk loud. Okay.
Strong voice.
Okay, will do. Thank you all for being here. As Dr. Gotterman said, Joycelynne Palmer, I'm at City of Hope, a former grad student in his program. And I was invited here today to present on a topic of my choosing. And so I selected this topic, which I think is pretty interesting and relevant, at least to the work that I do and the other faculty within the division. And it really, the talk is really going to focus on sort of the more recent evolution of clinical trials design. When I was here, I was an epi student. I was very interested in genetic epi. That was sort of the hot thing at the time. And then I took Dr. Susan Groshen's class. I don't know if I think she's recently retired or emeritus of some sort now, but I got a very sort of sharp interest in clinical trials. And so I proceeded with finishing my epi degree, and then picked up a position at City of Hope while I was finishing my doctorate here. So it's just a real pleasure to be back here. So I'll start with sort of framing the topic a little bit, giving some context, which Dr. Malcolm Pike always taught me to do. So I'll give you a little bit of background around sort of the treatment biology, just a little bit, sort of where CAR T cells are currently being used and what cancers, specific cancers, and then move into talking about some of the evolution within the clinical trial space, and then conclude with an example. And I can take questions along the way if there's something that is hanging you up a bit. Otherwise, I can we can take some questions at the end. Either way is fine with me. I can wrap up on time. I can conclude, you know, either deep or not in the example. So I've got some flexibility there. So just even before I jump into sort of describing the treatment and the biology that underlies that, I'll just tell you sort of where I live, because I think this is important for grad students and young faculty to sort of understand where biostatistics is now. When I was in school, it was genetic epi and biomarkers. Those were the buzzwords where everyone was spending their time and energy and working to get grants to explore and study. Now it's data science, it's machine learning, it's AI, where we have other data scientists and machine learners using a lot of the tools that we learn as part of our training as biostatisticians and epidemiologists So with that as the kind of backdrop there, I don't know if you can see my, you can't. So I'll just talk you through this. So I'm there in the middle. I'm the director of the Division of Biostatistics. I wear a lot of hats. I report up to Dr. Vadahi, who is a chairperson and professor of our Department of Computational and Quantitative Medicine. My division is actually one of 5 divisions. The other 4 up there in the left-hand corner, and you'll get a feel for the role that data science and computational biology and other quantitative fields sort of are playing in cancer research these days and other fields as well. So my colleagues, I work very closely with the Division of Mathematics and Mathematical Oncology and computational systems biology. We have a bioinformaticist that we collaborate with quite a bit. There is a director of health analytics, and then another director that oversees the space of mathematics for cancer evolution and early detection, which I know a lot of that happens here as well. There are 13 faculty in the division, about 7.5, 8 of those additional that are staff members, so statistical programmers and biostatisticians, staff biostatisticians. I have 1 data quality analyst because no data is good or usable unless you've got somebody really looking at it. And then, of course, every Biostat core division needs a program coordinator. So it's about 22.5, 23 FTE. So I'd say we're middle-sized, I would think. But we are growing in response to a lot of the growth that City of Hope is experiencing currently. When I came in as director in 2019, I reorganized the division to align statisticians with areas of specialization. One thing that I'm sure you all appreciate is that medicine and diseases are hard, and they're very deep now in terms of coming up with good solutions to problems that scientists and clinicians bring to us. And so it's in the literature where they sort of describe a bit now about the importance of aligning biostatisticians with areas of specialization. And so that's what I did here. And so these 10 boxes represent either clinical departments or areas of research at City of Hope, where we have dedicated faculty that sort of understand the nature of the challenges associated with progression, diagnosis, et cetera, in the various spaces. And so it's just a really nice sort of integrated environment where we have lots of rich opportunities for collaboration. Oh yeah, please, yeah. Ish, yeah. Uh-huh.
And I guess the other question is that you do, I'm sure you handle a lot of complicated data. Like there could be a lot of overlap. So how does that all
Worked out? Yeah, right.
Maybe you work closely with those folks and kind of share resources, or do you do your own thing?
Yeah, no, that's a great question. So I work more closely with the math onc and computational systems biology folks, as well as the bioinformaticists, the integrated genomics folks. We actually work on projects together, or we're named on grants together. So for example, in the math onc area, we work on projects looking at sort of the small example without getting too far into the weeds. I'll design the trial. I will develop the correlative studies, at least the analysis around the correlative studies, make sure we're powered sufficiently to answer the questions proposed. Then the math onc colleagues will come in and actually take some of those specimens and some of the clinical data and try to do prediction modeling through mathematical methods. To Russ Rockney, who is the director of that division, he's actually built out some elegant models looking at state changes using some elements from physics to, again, based on samples collected from patients and some preclinical models, and knowing the outcomes from the clinical trial will help to use those parameters and mathematical models from physics to predict. And so he and I collaborate quite a bit on grant submissions. We have to be really clear about sort of how his modeling is distinct from what I do. And so that's where we sort of feed on, or contribute to the project and sort of feed off of each other in terms of thinking through these problems. So he really gets deep into sort of what are the kinetics of the disease and what's happening with the patient. Of course, it requires a lot of measurements, longitudinal measurements to be able to do that. And then on the bioinformatics side, a lot of the RNA-seq single cell analysis, where we're trying to optimize treatment. Shiwei Wu, who is the director of that division, will take samples from patients, tumor tissue, blood, and evaluate sort of how the underlying biology, the genetics of the disease are evolving over time. And so we work really close together. Now, the two other areas, more collaborative. So in health analytics, Jim Lacey actually is now leading the teacher study. I'm forgetting the full name of it. Thank you, California Teacher Study. That Dr. Leslie Bernstein was actually the, I think, pioneer and architect of that. And so he's just continuing that work. And so our collaborations are just more of consulting. And then lastly, on the mathematics for cancer evolution and early detection, again, it's more just, is he thinking about the head, Dr. Tomasetti, is he really thinking about the problem correctly? And we sort of collaborate that way. He uses, of course, genome data, high dimensional data mostly. Yeah. So that's how we sort of all fit together and complement. And then this, I won't read all of this, and this is a very busy slide, I understand. But I just want to sort of point out how we're organized, again, to emphasize the importance of the connectedness of the respective areas and how clinicians, basic scientists, biostatisticians, how we all work together to answer questions. So those the 5 dark blue sort of rectangles in the middle there, those are our cancer center programs. So City of Hope is a comprehensive cancer center. We have programs that have plans, right? And we have statisticians that are named on that grant and then sort of assigned out to these programs. The layer down from there are these disease and modality teams. So all of our work at City of Hope is organized around disease teams. So there are regular engagements around the trial, whether it's an observational study or a prospective clinical trial, whatever these modality teams are interested in pursuing, we have statisticians that are staffed and allocated according to that structure that I just showed you on the previous slide, where we are all planted in these various disease and modality teams. And then the beauty of connecting the program to the modality teams where the work actually gets done is that we have opportunities to contribute to the science. And so the large bubbles or squares or shapes that are out to the side shows a sampling of what our contributions have been, how biostatisticians contribute our expertise to the questions that are being proposed in these areas. And so it varies, and I won't read it to you, but it goes from trials to prediction modeling to novel trial designs and new methods and mathematical modeling. So it's a very rich space. And it's this environment that affords me and the other faculty within biostatistics to engage on really amazing projects like this. And this slide covers the background of a key research focus area at City of Hope related to immunotherapy. So immunotherapy, CAR T-cell research isn't unique to City of Hope. Lots of cancer centers and medical centers are doing this kind of work, developing out new CARs to better target cancer. But just give you a little bit of background. And again, as to be sort of a good or great biostatistician, you've got to understand your subject matter. The better you understand your subject matter, the more you're able to contribute and add value. And so just a bit on background, so you have context. So cellular immunotherapy is actually an umbrella term for different types of treatments. So it covers cancer vaccines, autologous and allogeneic cells as part of a hematopoietic stem cell transplant, and then of course CAR T cells. So what are they? They're, don't have a pointer here, but what happens initially is you see the little patient there. The patient has blood collected, and that patient's T cells are harvested, pulled off, selected, and then genetically modified to carry chimeric antigen receptors. And then that receptor is designed to recognize and then destroy the cancer cell. And so this is targeted therapy. So for example, if I get to it, one of the examples that I'm going to show you relates to acute myeloid leukemia, where one of the mutations that is often seen in AML is a FLT3 mutation. And so at City of Hope, we're developing CARs to recognize FLT3 so that we can use the T cell to go and kill the cells in it, the cancer cells in a specific way. It's been shown as a promising treatment for blood cancers. Not that there isn't research going on in other places, but the real success first came in pediatric ALL at Penn with Dr. June, and then it's really just flourished from there. So that first indication that the FDA granted for the CD19 CAR. So this table here, to the right, to your right, is the list of CARs that are now commercially available, what their target antigen is, and then corresponding disease, and then who the target population is for the indication from the FDA. This list goes through, the last indication, at least on the table, goes through 2022. But recently, just in February, the FDA approved another cellular therapy for melanoma. So this is a really big deal for relapse refractory melanoma. And so it's just sort of showing the activity in this space, again, starting around 2017. Of course, the research was happening before that to present time. So with all of this activity in the CAR T cell space, it wasn't too long that clinicians, the scientists that were building these CARs, and the statisticians sort of understood that we had a different problem here, meaning the complications associated with CAR T cell therapy were not the same. The timing of the adverse events, the type of adverse events were quite a bit different than what we were seeing with cytotoxic chemotherapy. And so the little table on the left there shows some of these differences that we sort of, after running a few of these trials, became acutely aware of. So for example, and the reason I want to point this out as part of background is this is where we started to see that classic phase 1 trial design was just not going to work. I mean, you could build a justification and use a standard traditional phase 1 trial design, but was it the best design? It became a question, and here's why. So I'll focus on toxicity just for the sake of time here. So regarding toxicity, in cytotoxic chemotherapy, the toxicity rates, the grade and the number is really high at higher therapeutic doses, and there tends to be a very narrow therapeutic window. So the range at which the doses at which the chemotherapies works is a bit more narrow, and toxicity is cumulative after repetitive exposure, repeat challenges. And so this was what we were walking into CAR T cell therapy sort of with in sort of the back of our minds. This was our understanding of what we thought, what we knew the toxicity profiles would sort of be like. But what we noticed with CAR T therapy was that toxicity was very low. And because the treatment is targeted, we weren't really surprised by that, but we were sort of concerned about other biologic effects of the treatment. but we didn't really see that. We don't see, and that's still true today. We don't see a lot of toxicity in terms of, there are some specific ones which I'll talk about in a second, but in terms of the broad span of adverse events, we don't see it. And practically, there's no real lethal dose. So you don't, like with chemotherapy, you escalate and escalate till you can't anymore. Patient starts to fall apart. That's not true with immunotherapy or CAR T cells. And to date, because CARs are all specific, right? They're targeted therapies to a given disease. We at least I don't feel that we have a very well-defined dose effect relationship just yet, for two reasons. One is it's still fairly novel relative to chemotherapy, studying that for a long time. And then the other part of that has to do with sort of understanding how the CAR behaves once it's actually in the body. There are potent CARs and less potent CARs, which I'll get to in a moment. And so this was sort of what we began to see back in, well, not that long ago, but I would say probably around 2015 or so. We started to hear in the work that was coming out of some of these centers that were running phase 1 studies that this was sort of the behavior. And so this made us sort of scratch our heads a little bit. Once we started, we had a few more trials under our belt. It was very clear that not only were we seeing a lower number of adverse events as in terms of severity, but we were seeing sort of these unique toxicities that we hadn't seen before in the CAR T cell space. The two big ones are cytokine release syndrome, and the second is what we call ICANS, the immune effector cell neurotoxicity syndrome, which is actually made up of, it's called a syndrome because the neurotox is actually a few different things that it can manifest in a few different ways. Patient can have seizures, motor findings, speech issues. These were all unique to CAR T therapy, and lots of papers are out now and sort of describing that and coming up with different grading system for CAR T cell therapy. So we decided that we wanted to sort of look at what was happening with cytokines, because they appeared to be the key driver of the cytokine release problem that we were observing. And so the very last, to the left on figure 1, the very last graphic there at the bottom shows the pattern of peak and response of the various cytokines in response to the patient receiving CAR T cells. So you can see these cytokines all have different roles and different behaviors in terms of regulating immune cells, but they all sort of peak, and the length of their peak response, or before it starts tapering off into the tail, is a bit different. And so we began to sort of get interested in this because we realized, in addition to the response of the cytokines once the patients receive the CAR T cells, so again, the cytokines turn on the milieu, the environment of the patient to receive the CAR T cells and have them sort of do their job in targeting the cancer cells. While all of that's happening, we're sort of happy about that because we know the CAR is invoking a response. But on the other side, it has these adverse events associated with it. So how do we manage that? Well, so we started, oh yeah. No, but they're associated. They're correlated. There is a relationship, but you don't see like the height or magnitude of a cytokine being associated with the severity of their ICANS grade, for example. But when you have one, you typically see the other, but it doesn't necessarily mean that one drives the other, or if one's higher, the other one's higher or lower. Yeah, sure. And then thank you for that. And then in the top panel there, a figure one, you can see that ICANS actually, we realized when we started studying this, that out from the CAR T cell infusion that you start to see the sort of delayed effect of the neurotox that happen. So they don't happen at the same time. But this was unknown when patients were first treated with CARs. And it caused us to sort of, it's still within this short period of about 28 days, but with sort of the turning on of the cytokines and this neurotox that was happening, we realized that we needed to sort of do something about this, or at least keep an eye on it as it relates to trial design. So how this sort of began to influence the way that we designed phase 1 trials, I give you a sample schema here. Just made this up, and I can't point to anything, but I guess, okay, so I'll just say that the package of the CAR T cell therapy starts with something called lymphodepletion, where the chemo, oh, thank you. Oh, okay. Okay, for those online, I'm sorry, you can't see where I'm, yeah. So the CAR T cell therapy actually begins with something called lymphodepletion, where the physicians use 2 agents, fludarabine and Cytoxan, to create space in the bone marrow compartment to allow the CAR T cells to come in and work their magic. There is all of this, which I won't go over today, but there are considerations when you're designing one of these trials around manufacturing. So before, when we had, my early sort of clinical trials were in the transplant space under Dr. Joycelynne, and we didn't concern ourselves with hematopoietic stem cell transplants on this prep area. This was sort of behind the curtain, if you will, for the biostatisticians, the harvesting of cells and all of that. That just happened. And then we got the date that they were going to get the cells and we were ready to go. So one thing that we didn't, as biostatisticians in designing these trials, we didn't know how involved we should be in this piece. It turned out that there was some guidance that was needed in terms of counting evaluable patients and such, and evaluate when you're looking at this as an entire package. So I'll just leave it there because there's a whole nother area of study that discussion that I could go into, and I won't. So the lymphodepletion originally was also part of this package because we early days didn't know what the appropriate lymphodepletion was, what the optimal lymphodepletion was, whether or not we needed it. These were all questions, and so the early studies did not. Were really focused, the biostatisticians and the clinical team really focused on understanding the role of lymphodepletion as it relates to toxicity, et cetera. So this is the schema, the classic schema. We for a CAR T study, and we are again often discussing lymphodepleting chemotherapy, how much, what to give, although those questions are mostly settled these days. But then this persistence activity endpoint, as well as disease assessments at day 28, became really critical, which prior to the CAR coming into sort of this aspect, this type of immunotherapy, I didn't quite understand the relevance and importance of persistence. And I learned very quickly that the reason that we needed to have the persistence assessments done was that these treatments are incredibly expensive, right? So if the CAR is having no activity, maybe we're not seeing a cytokine response, maybe we're not seeing any ICANS, there's no toxicity indication that something's going on, and we don't see the cells, the CAR T cells expanding on day 28 or by day, or still around by day 28, maybe we need to go back and reevaluate the CAR construct, right? So the physicians and scientists started telling me, hey, Joycelynne, you've got to focus on persistence also. We really care about that. And I and other statisticians were saying, what are you talking about? This isn't about efficacy at all. I just want to know if it's safe or not. And they said, no, really expensive, and we need to re-engineer our car. So you've got to factor this into your design, right? Phase 1 study that's looking at an efficacy endpoint, including disease response. Hadn't done that before in making decisions, particularly in a dose finding first in human study. I'll continue. For sake of time, I might just go over this because really the point here isn't to rehash a 3+3 design or an accelerated titration design in the phase 1 space, but rather to just point out that this is our history in biostatistics, right? For those of us that work in the clinical trial space, these have been around for a long time. 3+3 has been around for a very long time. There are lots of derivatives of this design these days, but this is sort of where we lived in the chemotherapy space. So when the clinicians were coming to me and saying, hey, you've got to figure out how to factor in persistence, these are all toxicity driven, right? And at best, go to the next slide, we move away from the algorithmic designs and move into the model-based designs. These don't factor in response at all either, right? This is all, again, based on prior understanding of dose, what we think the DLT rates are, using information that's accumulating on patients treated and having that inform our prior to make decisions about where to assign the next patient. This is the space that we were in. I put history ? Just because we do still use some of these designs in the CAR T space when appropriate justification is there, but this is what we had to work with. So then the FDA came to save us a little bit because we were struggling with figuring out how to make these more elegant designs with the tools that we had at the time. So this guidance came out March 2022, and it helps us as trialists understand how to get our trials through FDA review, because these are all, at least the constructs at City of Hope that we build, they're all first in human, they all start there. How to get them through the FDA. What is the FDA thinking about when it comes to this idea of not pushing a CAR to the maximum tolerated dose, right? Rather using toxicity and response to come up with something that looks more like an optimal biological dose or effective dose. So they came out with this guidance after much, back and forth, and the research community begging the FDA to pull through for us with the document that gave us some guidance. So where they ended in this migration from, an MTD, maximum tolerated dose chemotherapy sort of mindset to an optimal biological dose mindset, they in that move they gave these guidance, and so I've highlighted just a few of them here. So the FDA believes that we need to find the biologically active dose, and they want us to determine a range, not a dose, because they understand that the activity around the dosing can vary. They think of it as a biologically active dose, and we have to have measures to support that it's biologically active, in addition to saying whether or not the patient responded or not. And if you want to get really fancy and looking at persistence and the relationship, so there is a correlation between cytokine release levels and persistence of the CARs. If you wanted to use that as a parameter and evaluation and determining your optimal biological dose or effective dose, you've got to put that into your design as well. But for many of these cell therapies, there are practical limits on the dose because a lot of these products are manufactured from the patient. They're considered autologous products, and these patients are sick, and we're trying to mobilize T cells from their bone marrow, and they have, in most cases, a blood-related cancer. What dose we can go up to may be capped by what the patient can produce. Another thing to consider. Response rates, and here's the really important one. And going back to that distinction I made between immunotherapy and cytotoxic chemotherapy, we've demonstrated that the response rates really become flat at a certain point over a range of doses. So I think I have a slide in here on this, but I'll just say it now. With cytotoxic chemotherapy, if your x-axis is the dose and your y-axis is the rate of adverse event or response, with cancer chemotherapy, as the dose increases, toxicity increases, right? Or dose increases, toxicity increases, but then the response increases, right? So physicians want to give as much of the drug as is safe to do to get the patient to respond. In biologics, the CAR T side, immunotherapy, not the same relationship. Toxicity may continue to increase, depends on how potent your CAR is, but once you get to a certain dose, because it's targeted therapy and you can saturate the tumor space, you start to see the response level off. So it doesn't matter how far you push the dose, you're not going to see an increase in response because of the saturation that happens. So this is a really, really important thing that when the scientists were talking to me about this, just to be honest, I did not understand what they were talking about. I've never heard this before. It made no sense. But when you start to look at the data, it becomes really obvious. Okay. And then more guidance documents came out, and these are, if you're interested in this, I would Google them and read them. There's a lot of legalese in there, what reads like legalese and government speak, but there's some really good nuggets in there as it relates to sort of what maybe some of you might be able to have a word on. So where does this get us? So what's happened, so there's 2 points I want to make here. One is, which is more of a global kind of side comment. The clinical trial space is moving now from what I would consider, like when I was here, the more traditional design where you have these very formal demarcations from phase 1 to 2 to 3 to 4. I never did any of these, only did one of these. I've spent most of my career in this space, where there are these designs that happen. Maybe they're first in human, you determine your MTD, they're considered dose finding studies, you do some pharmacokinetics on it to figure out where the drug is going, how it gets expelled from the body, and then you move it on to a phase 2, right? This is sort of the space that I walked into back in 1998. Now, the world looks more like this. And I think this is an important point to make. And if you review some of the FDA documents, sort of get this from their writing, that this is, you could sort of make this leap, even though this figure, I'm not sure, is in that document. So where are we now? It's more of this non-traditional, what I would call non-traditional sort of state space as it relates to clinical drug or biologics, immune therapy, cancer-directed immune therapy. So now what's happening is we're instead looking at safety, of course, tolerability, so things that may not register as a severe adverse event, but it is making the patient sort of be intolerable to receive the treatment, is informative, right? Traditional mindset was, if you don't have a grade 3, 4, 5 adverse event, you keep pushing the drug, and you don't really pay attention to quality of life because you're trying to save the patient's life, right? But now, with targeted therapies, we're more interested in tolerability. There's a lot of patient-reported outcomes work happening in that space now. And we're also looking at feasibility. That manufacturing part that I showed on the previous the treatment schema, sort of made up one, that is one of the considerations when we look at the overall package in designing a trial. Is it feasible to get the patient in, determine their eligibility, harvest cells, manufacture, re-engineer the T cells to give them back when they have a cancer that could just blast through in 30 days, right? So is it feasible and do we have a way to keep the patient their cancer contained while we go through these steps? So that's sort of a new aspect of this. It's targeted therapy. There may be a limited population. Some of these mutations are on the rare side. As I mentioned, the dose finding is really about optimal dose. We look at activity now early. That's just accepted and in a lot of cases required. It really requires innovative trial design. And in some cases, we I don't know if the word is exploit. We have to provide justification, but we really take advantage of this expansion cohort concept to get good estimates of activity, whether it's a clinical response or something related to the persistence of the CAR. This never happened before. So you see larger phase 1 studies now. Even in the CRM time, this was being pushed and people and physicians like really pushed back because those Bayesian model-based designs require a lot of patience. But now the clinicians and basic scientists are really on board with this because they understand the value of estimating response early on before they take this very expensive product forward to phase 2. They want to make sure they've got that CAR as good as they believe it can be. So this is different, right? We often have now phase 1 studies that are very big, like 40 patients with like 3 dose levels. It's still 40 patients. It used to be like 18. And again, it's the idea is to get some early signals to figure out what's happening to the cancer and whether we're justified in proceeding. So I think I'll skip this slide. I think I talked about that. And then this is just a graphic of what I was describing about the relationship between toxicity and response. So this is what the cited in graphic form, this top right figure here. What I was describing is, the therapeutic window is really between here. You start to get early response, but you have to push the dose to get something better, but it's not without increased toxicity. Whereas on the CAR T side, you have this sort of more narrow, I would say. Depends. The behavior is just a little bit different, so that you have to get, this is the minimum, what you're interested in CAR T now, again, looking at activity as well as toxicity, is you want to establish what we call the minimal effective dose across the dose ranges explored, and then it says MTD up here, but really we tend to find the OBD somewhere in here. Ooh, there's a little delay here. I think I've gone over this, so I'm going to want to make sure I have enough, at least a few minutes to talk about this. So here's what the response has been by biostatisticians to this problem or challenge of incorporating activity into a phase 1 study, where you're making decisions about where to assign patients and their dose, not just on adverse events, but also on activity, which again can include a few different things as part of that endpoint. So this slide summarizes, and I have the reference here, a suite of what we refer fondly to as innovative trial designs, and this is for phase 1 and phase 2. Ying Yuan at MD Anderson has just been a trailblazer in building out a suite of designs, which you may have heard of by now. They're called Bayesian optimal interval designs, BOIN designs, and he's got about, I'll just say maybe 10 if I'll count all the variations, which has been just a real workhorse for us. And he has a lot of great papers to show its performance relative to some of the earlier methods that we use, like the tight CRM. So I won't read this to you, but just want to sort of make the point that the model assumptions that we're now considering in the CAR T space, again, factors in toxicity as well as activity. And I don't see the one that I'm going to talk about in a moment, but this is just to show you that this has sort of been the response by biostatisticians. And the other thing that I just want to say really quickly is one of the impediments to using like a continual reassessment method, some of the Bayesian designs for phase 1 trials, is that there just wasn't good software available. So what would happen is when a patient came to be treated, the biostatistician would get called to ask, paged. I mean, we had pagers to, I know, it's like, wow. And then I call Joyce. What do I do? So we would get called, and then we would be in the center, in between the patient and their treatment, right? Because now there's a patient that's been screened and is eligible and ready for treatment. And now I've got to tell the team where to treat the patient, what dose. But I can't do that alone, right? I need data. So then I'm looking at data. What if the data's not there? Then I have to call the data manager and ask, where's the data? So it just became this logistical nightmare a bit. And people, sort of, this was a while ago, but I mean, you could get called like at 5 o'clock on a Friday. I mean, and you gotta answer the call, and you gotta help make the decision. So it just became really cumbersome to sort of understand where to go. And there were too many people that had to be in the middle of that process. The CRM folks, the authors of that, are starting to build out more, and you can buy software that helps you get through that. And I have no conflict of interest, no affiliation with Dr. Yuan at MD Anderson. I get no royalties or anything from the Boyne suite. But I'll tell you, the wonderful thing about the Boyne package is that it's all there for you. What do I mean by that? You go in and you specify your operating characteristics around sort of your threshold DLT rate, what your target response rate is, because we need both of those in CAR T. And you enter in some parameters to check how well it performs given X number of patients and X number of dose levels. And it spits out this beautiful summary of findings, including a spreadsheet. And it depends on how big your study is, but it can go out like to 12 Excel spreadsheets with all the scenarios. So it's there. I mean, of course, you don't put all that in the protocol, but it's the locked record for the design so that when the trial is running, people know what to do. So we've made leaps and bounds on the incorporation of activity and the tools that statisticians need. And honestly, the team needs to run the trial. So I'll just, looking at the time, so just I'll wrap up. So what do the objectives look like? I like to see examples. This is, there's gaps in the text here just because that's where you would plug in your CAR name. But this is how the objectives have changed. So originally, we would just have the first primary objective, right? It would be 1, and that would be to assess safety and determine the MTD. That was it. That's what we did. But now we also, as a co-primary endpoint, and this is what some of the designs I was just talking about do for us, they allow us to evaluate the anti-tumor effect. This is a made-up leukemia example, so it would be the anti-leukemic effect. And then, any good statistician, because there's lots of things that we could look at to evaluate efficacy, good statisticians have to tell you what you're looking at for efficacy, right? So here we were using complete remission, CR or a CRI, that's a complete remission with a incomplete recovery of the bone marrow. And then you have to tell the time, so it's the first 28 days. And the beauty of CAR T therapy is you can get responses early. So this is not unreasonable to look for a clinical response early. And then some of the secondary objectives here include the usual suspects: overall response rate, PFS, overall survival. And then here's where the fun is. It's in the correlative studies, because the correlative studies really help us to understand what that CAR is doing, right? What's going on under the hood. So we can look at, let me just pick one. We can, let's see, maybe down here, and I try to count for those that are on Zoom. I can't see it from here. Within each immune cell population, so it's 1, 2, 3, 4, 5, 6, the 6th bullet down, we can conduct what we call differential expression and pathway analysis. This is where some of the informatics comes in, Jim, on this one, to explore the gene pathways associated with response, so we can get down to that level. the third bullet up from the bottom, we always profile the cytokine levels, graphically, because that's the way it's understandable over time. And in the presence of cytokine release syndrome. So of course, if the cytokines get too high, that's how CRS kicks on. So, and then there are a lot of analyses of the actual bone marrow in this case, because that's leukemia where the cancer is, to see if we see CAR T cells in the bone marrow. So we do a biopsy at day 28. We're looking for response. We're also looking to see if there are CAR T cells there, and of course in the blood circulating. And then maybe the last thing I'll say here is this is sort of what a study design section looks like. It includes details about what type of design you're using, obviously. It's focused on optimal biological dose. Of course, we have to talk about the number of dose levels. In the Boyne design and with most of the Bayesian designs, you have to consider all dose levels, even if you don't intend to or think you'll go down. And you have to describe sort of what the treatment is and how many infusions they're going to get. You have to talk about whether or not there'll be intra-patient dose escalation. At least for my studies at City of Hope, we're not doing that yet. But the FDA likes to make sure we're clear. And then I guess the last thing I'll say is with this utility-based Boyne design, there are 2 stages to it. And if you want to look it up, the first stage only factors in toxicity according to these probabilities. And this has to be sorted out with the clinician as to what they think is tolerable. Here, we set the threshold at 30%, 95% probability to be concerned. And then once we've ruled in the dose levels that are considered safe of those that are being tested, we then flip over to stage 2, which is here. And this is where we begin to look at the toxicity efficacy trade-off according to these operating characteristics here. And in the end, the optimal biological dose this is kind of the punchline, is defined as the dose that is admissible and has the highest utility. So this means safe and is effective or active. So I'm looking at the time, looks like I have 3 minutes left. I'll probably hold off. I'll probably pause there. I have more slides, but I think for sake of time, I will wrap up there and see if there are time for questions.
On your list of forward, I noticed trial. Is that something that you use to sort of evaluate, whether the CAR T cells are too old, or is it something that you can use beforehand to estimate maybe the efficacy of it?
Yeah, that's a great question. So it's the first thing that you mentioned. We're looking to see how well the CAR is performing. And so what cells are being engaged or amplified is where we're focused there. There's not a lot of real hard statistics there because there's so many things to measure. And so we just use it more as a qualitative, measure. But I will say that if for really potent CARs, some of those cells tend to amplify or increase way more than what we want or expect or what is considered normal. And so that'll cause us to maybe dial back some of maybe the dose. And when we do get really potent CARs where, we want to go back to the FDA to sort of, because, they can see everything, right? Across the US, all the INDs. They are very good at making recommendations on how we might modify the CAR to help to get the profile looking more like what is reasonable. Yeah, that's a great question. Right.
genetically or other
Right.
years might measure.
Right. Yeah. I think what we usually do, particularly because again they're targeted T cells, so like a CD19 or a FLT3 CAR. We've realized that the level of expression of an AML cell that has the FLT3 marker is really important, and because it's that saturation issue. So if we have patients that have very high levels of FLT3 expression, the CAR may not be able to find its way to do much, right? And so then is the question, do you radiate first, or do you give them some chemotherapy first and then take them to CAR as a consolidation? And that's sort of what's happened, because I think to answer your question directly, it's really been about the level of expression of a certain marker for that disease that really guides us on, do we think the patient will benefit or not? Now with phase 1 studies, it's usually all comers, but we use these correlative studies to sort of sort out if we really, to your point, want to really seriously figure out who this therapy might be good for. Maybe the eligibility criteria wouldn't include someone that had high levels of expression. Again, you get them into some treatment first, try to lessen the tumor burden, and then take them to CAR. The other thing we've done is we've done CAR to try to consolidate a response. We wrote a CLL, an ALL paper with CD19, where the point of the study, and then what the paper shows is that if you can consolidate their ALL, like get them into a deeper response. So these patients are coming in with minimal residual disease, some, not high levels. If you can use the CAR to consolidate, so have the CAR target those whatever residual cells are, get them into a deeper remission, you can take them to transplant, particularly cure them. And so now CAR is becoming like every other tool in the toolkit. It's not a one and done unless it's Gleevec, but it needs to be added with something else. So, but I think they know that now. That's been one of the big ones, is how much disease, how much expression of the marker is happening in the patient. Yeah. Yeah.
An early phase trial, you don't have a comparison.
Right.
So usually for the efficacy purposes, use historical data, or what's a typical approach for assessing efficacy in that situation?
Yeah, right. So when we try to come up with these response targets, this is 75% for this one. We use historical response data for the patient population that we're targeting in the study, so that I've had that where, not pointing any fingers, but I'll get references and I'll say, wait, those aren't our people. I need another one, right? So we will often use historical, but then what we also do is because of the cost and the time and the commitment of the patient, we will aim high. So let's say in relapsed refractory leukemia, a lot of the targets are around 30% response after the first cycle. But because again, this is more than that, we set it higher, so it becomes a question of what's clinically meaningful as opposed to what the historical rates are telling us. And the good thing about this design is another great point to bring up here is that we're targeting a very high response rate here, but if we don't get that doesn't mean the study failed. This isn't that game. Phase 2 studies, when you do interim analysis and you don't hit your mark, it's like, oh, stop the trial, start over again. Here, no, it's just guiding where you treat the next patient. So if you haven't hit that threshold and there's no toxicity, the design pushes you up. So yes, we typically use, and we do have to sort of cite in the background sort of what's been done in this population, as the setup for why we think this therapy is needed. And then we justify the 75% as something that's clinically meaningful, clinically impactful. And the FDA allows us to do that. Yeah.
Dr. Lee has a hand raised.
Okay.
Bing?
Hi, Joyce. Nice talk. And yeah, I have a question, practical questions. When you work with the clinicians, physician, they are used to the like 3+3, the traditional trial, right? So when we like introduce like Bayesian Bayesian design, sometimes they have some kind of a fear, like, oh, this is a involve probability, all those things. Is like we don't understand. We probably will rely on you most. But to us, we think at the beginning we have those detailed outline, right? Blueprint, everything is it's even straightforward. They have those things. They can be recruit patients based on their all those parameters. So in your practice, how do you convince them this is a better design? I have those experience like, oh, I need to work hard to see, okay, this is because in the boring design, we always say, okay, equivalent. In this scenario, we equivalent to 3+3, which can the dilemma for Bayesian statistician is like always need to like convince them, like, see, we are equivalent to you. Then we are some scenario better than you. So maybe you can share with us how you convince the physician this is kind of better for you, not like put more burden on you.
Yeah, that's great. The pushback, great question. The pushback that I received when I started to introduce these designs was the expense, right? Because as you know, any Bayesian design is going to require more patients, right? So there was a big sort of resistance to it because they would look at the sample size number and like, okay, on this study, they would need 9 patients, right? Or 3+3, 3 and 6, you're done. And I would come and say, well, no, I need 18 or I need 20 or 23. So what I did, and you'll see this on this was the first sort of baby step. First thing I did was the Boyne utility, the Boyne suite, I think it's trialdesign.org, I think is the website. I have a bookmark, so I don't remember. There is an option in the Boyne and the UBoyne to select as part of stage 1, a 3+3 run-in. So remember I mentioned that, and I know you know this, maybe for the sake of everyone else on the call, stage 1, as I mentioned, is just exploring the toxicity space. Are these doses safe? That's it. So what I started with was, okay, let's do for stage 1, where we're just exploring the dose space and evaluating toxicity. Let's use a 3+3. I just give them that. And then everyone feels very comfortable, right? But then when we switch to stage 2, it switches over, when we move to stage 2, it switches over to the utility-based Boyne design, where we're now looking at my probabilities down here. So any dose, when we switch to stage 2, where the toxicities are below our acceptable levels, they get factored into an evaluation of response. So for the first piece of it, because the other thing, I'm going to try to be concise here. The other thing about the some of these UBoyne designs, if I use CAR T-cell persistence as my activity endpoint, because they think that there's just makes biological sense or scientific sense to do that. I also am putting pressure on the people that have to run those assays, right? So it made everyone happy to say, okay, I'm just for stage 1 going to look at toxicity. I'll do a 3+3 run-in, but then when we get to stage 2, we will begin assigning patients according to these operating characteristics. And they seem to agree with that. For those that are just starting to try to get their minds around this. Eventually, after a few of these, the clinicians get to a place after they've done a couple and they see that nothing breaks. They get to a place where they understand these toxicity response curves. If I can get there. And they understand that it is absolutely essential to understand when response is also important to characterize sort of what the response looks like after you get above or at a certain level. So I think that showing them, and I don't know if you're dealing with CAR T cells, so I'm maybe not directly answering your question, but at least in this space, because it's expensive, because there is a need to sort of understand underlying activity, they will give me the patients and let me work around with probabilities for two reasons. One is they have better confidence about their estimates going into phase 2, which they really want, right? They don't want to see something that looks promising and it doesn't, you know, hold up when we look at more patients. And then the other reason they buy into it is I'm only going to if their CAR T cells are effective, or let's say their chemotherapy is effective, they're going to want to get some good indication of activity. And so that estimate that you'll get, along with the complementary toxicity profile, will help the physician sort of understand the whole package. So I guess, I mean, I know there's a lot of words to try to answer your question, but I think this idea of, if you wanted to use the BOIN suite, I'm pretty sure the BOIN, not the UBOIN, but the BOIN has a 3+3 option. You might want to try that. Use that for stage 1, and then the larger BOIN after, where you're actually looking at coming up with a more accurate estimate of DLT.
Thank you. Thank you. Yeah. Thank you.
Thank you. Thank you for having me.
Thank you, online.